My name is Benjamin, and I'm data scientist at Buer Crop Science in Monheim, Germany, one of 3 divisions at Buer, besides the pharmaceutical and healthcare divisions. For 10 years, I'm working in small molecule department, and together with my colleagues, we are hunting for novel and sustainable crop protection products. My background is in plant physiology and molecular biology, and that helps me to bridge a gap between wet lab researchers and data scientists and to understand how we can enable wet lab researchers to work with multi-omics data. At Buer, we have a very encouraging and ambitious mission: health for all, hunger for none. As a life science company, we have the unique opportunity to leverage this mission with our core competencies in healthcare and agriculture. For us in crop science, this means to focus on regenerative agriculture and to restore nature while producing more for growing world population. In small molecules in particular, we also recently announced the crop key strategy for sustainable crop protection. Our goal is to unlock the future of sustainable crop protection as with a crop key strategy. One key aspect is a target-based discovery comparable to pharmaceutical approaches. This includes in-depth physiological understanding of the pests we would like to stop feeding on the crops so that we can target them very specifically, followed, of course, by a detailed characterization of our assets. This is done by biochemical and computational biological approaches. Data is key here to make informed decisions and to advance our scientific achievement. With the advent of AI and systems biology, high-quality data is indispensable for us, and we must make it fair to our drug discovery experts. So make it findable, accessible, interoperable, and reusable. This enables us to identify patterns, trends, and correlations that are critical for the development of new products, improving our existing products, and to understand complex biological approaches. We initiated the search for a data management platform to complement our crop key strategy, which places emphasis on target-based discovery by leveraging systems biological AI to deepen understanding of biological systems. Through the integration of omics data and advanced analytical technologies, we seek to drive innovation and develop more effective and sustainable products for our customers. This requires a robust data management platform that could support our endeavors and enable seamless interaction with diverse data sources while adhering to FAIR principles. Our goal is to establish a state-of-the-art data environment that offers flexible, accessible, and integrated FAIR principles, facilitating interaction with the data. We aim to enable seamless retrieval and manipulation of the data, ensuring high-quality accessibility for AI and systems biological research, while making data and insights readily available to our experts. The ideal solution would prioritize unified data access, customizable views, cross-platform accessibility, data security and compliance, scalability, and of course performance, all aligned with the FAIR data principles. This approach will empower us to leverage our omics data dynamically and efficiently, fostering a cutting-edge data landscape. Before Genestack, our omics data was in a fluid state awaiting structure and organization. There was no consolidated view of available internal studies in small molecules, resulting in a lack of comprehensive insights, and access to omics data was limited mainly to individual project teams. The annotation of omics data was partly inadequate, lacking the necessary structure for efficient analysis. These limitations made it difficult to retrieve meaningful insights beyond the scope of individual projects and hindered the potential for comprehensive and integrated data analysis. We first met Genestack in an academic industry collaboration where Genestack was used to manage and transfer the generated data. To identify the best solution, however, we defined criteria for the ideal omics data management solution jointly with our experts in small molecules. Our colleagues in open innovation and strategic partnership supported us to identify a number of potential solutions, including Genestack that we investigated carefully. We decided to choose Genestack for an extended proof of concept due to its flexibility and the integration capacities, as well as interoperability with our systems. The platform allowed us to integrate our existing tools, solutions, and workflows to provide tailored access to our omics data assets. With Genestack, we can in future organize, analyze, and derive insights from large and diverse data sets efficiently.